It is my honor to give an oral report on the ILP conference. I'm Wen Jenny, come from the University of Electronic Science and Technology of China. Emotion regulation is an essential part of our daily life. To effectively regulate emotions, different types of strategies may be adopted. And in one of our recent studies, we recorded three typical strategies: distraction, reprisal, and suppression, using fMRI to investigate the neural mechanism of emotion regulation. We found the reprisal strategy causes stronger activation in most parts of the frontal cortex, including the bilateral inferior frontal gyrus. Moreover, from the functional connectivity analysis, we observed that from the right FG, which served as a seed region, to the right DMTFC, only the connectivity under reprisal condition was negatively correlated with reprisal effect. That is to say, the increased connectivity was associated with more success of reprisal regulation. In another study, we used the emotion regulation questionnaire to measure the habitual use of reprisal strategy, and we applied the resting state functional connectivity analysis to explore the relationship between FG-based functional connectivity and the dispositional use of reprisal strategy. Interestingly, we found that FG-based functional connectivity was positively correlated with habitual reprisal. From the two studies, task-based fMRI and resting state fMRI, we can see the key function role of IFG in collaboration with other regions to facilitate a better emotion regulation of reprisal. To verify the core role of the right IFG in reprisal strategy, we then did a tDCS study. 20 participants were recruited to do the emotion regulation task, which consists of a view block and a reprisal block. In order to avoid carryover effects caused by the reprisal instruction, the order of the blocks was fixed, with the view block followed by the reprisal block. All subjects received 2 online tDCS stimulation sessions, with a time interval of 1 week. And the anodose stimulation session and the sham stimulation session were counterbalanced between subjects. For anodose stimulation of the FG, the anode electrode was placed over F6 according to the International 1020 EEG system. The cathode electrode was placed over the FP1. In the active stimulation session, a constant current of 2 mA was started 4 minutes before the task onset and prolonged for the entire task. While in the sham stimulation, the same current was started 4 minutes before the task onset but lasted for only 1 minute. The results showed that, for the rating of negative emotion, there was a significant effect of condition and a significant interaction between condition and stimulation, but there was no significant effect of stimulation. From picture B, we can see the emotion regulation effect was greater in the active tDCS than that in the sham tDCS. These results indicate that the anode tDCS over the right FG can facilitate the capacity of emotion recognition. That's my report. Thanks.